Okay guys, since the MCO will be extended, so I guess we need to record more lectures. Okay, so for the first part of the lecture is actually finished yesterday, which is the Professor Hasanah part, and this is actually Dr. Hanan's part, which is about fermentation. After I discussed with Dr. Hanan, he agreed to let me give you the lecture for this part as well. Okay, this slide initially contains about 40 to 50 slides, but I simplified them to about 15. Because this is just the introduction and I don't think you need to know very specific about the pathway that involves in the fermentation. Okay, so for this part, as you know, fermentation is very useful, especially in food industry. It can increase the enzyme content that can help you absorb food better. It can also provide you probiotics for the gut health. Fermentation can also increase the safety of the food. So instead of the spoilage bacteria, what actually growing inside your food is the beneficial ones. You can also preserve your food, make your food last longer. For example, if you put the fermented bacteria inside tempeh or kimchi, it can last longer compared to traditional storage. And finally, it can increase the nutrition. For example, in soybean fermentation tempeh, it can reduce the amount of anti-nutrient and increase the availability of nutrient. Okay, so this is the topic outcome. You can read it by yourself. Okay, so the background of the fermentation is actually the definition of fermentation is very wide. I don't think there is one definite definition. But for me, as long as the process include the any microorganism, it can be considered as fermentation. But certain definition of fermentation can also be done for enzymatic fermentation. For example, if you ferment the coffee or chocolate to develop the browning color without the involvement of any microorganism, it can also be called as fermentation. So, like I said, the definition for fermentation is very broad. Okay. So we can take a look at the first definition, which is any process involving microorganism or any biological process that occurs in absence of oxygen or very low oxygen amount. So if the microorganism is using oxygen, we call that as oxidative respiration instead of fermentation. So remember, fermentation must occur without the presence of O2 or very low oxygen. Food spoilage, I think, can be considered as fermentation as well because it falls under this definition. But because food spoilage usually produce, not usually, food spoilage produce harmful food or non-edible food, I think we cannot call them as fermentation anymore because fermentation produce a good food for the consumption. So the background is fermentation is energy yielding, means that it can produce ATP, although it is lower than the respiration. It involves the degradation of carbohydrate. So if the substrate is protein or lipid, I'm not very sure, but I think it can still considered as fermentation, but people tend to call this one as putrefaction and lipid as lipid degradation. But if we consider the use of microorganism, sometimes we can still use the word fermentation if the substrate is protein or lipid. Okay, fermentation is not involved when you have oxygen, like I said to you earlier. Okay, so this is the equation. So for anaerobic fermentation means that you don't have oxygen. Usually you will produce ethanol, carbon dioxide, and also low amount of ATP. If you use this one, usually true for human, but if in microorganism, they can produce much more things compared to these three things. Respiration, under enough amount of oxygen, you can produce CO2, H2O, and also more ATP, but no ethanol or lactic acid. Biochemistry point of view, the definition is you must use carbohydrate or sugars. That's why, like I said before, protein and lipid is probably not fermentation, and it can be convert or oxidized to other metabolites. For example, acids, many types of acids, gases, for example, CO2, and alcohol, for example, ethanol. And also, you must release the energy in the form of ATP, although the amount is very low. And you must not have any electron acceptor, which is for human usually oxygen. For bacteria, it can be sulfur and so on. So this electron acceptor is actually present in electron transport chain. That's why we need to breathe in oxygen. Okay. Modern definition extends this to be microbiological formation of smaller organic molecules, whether anaerobic or anaerobic. So sometimes, even though the things happening under the presence of oxygen, it can be considered as fermentation for certain people. So like I said to you, the definition is very broad. So probably we need to always keep up to date about what is the true definition of fermentation. Okay. So there are three major glycolytic pathways. Glycolytic means glycolysis, or the breaking of sugar, found in different microorganisms. The first one is AMP, which is the most famous one. Second is phosphocatalysis pathway, usually produce lactic acid and ethanol. And the last one is AD pathway. Usually they involve in the formation of alcohols. Okay. There are several types of fermentation. The first one is lactic acid fermentation. It can be homo, which means that it can produce exclusively lactic acid, or it can be hetero, means that apart from lactic acid, they can also produce other kind of acids. Okay. Other types of fermentation can be mixed acid fermentation. Okay. Mixed acid fermentation is actually very similar to hetero, but mixed acid fermentation using EMP pathway. Heterolactic fermentation usually use phosphocatalysis pathway. Okay. Acetic acid fermentation means that the bacteria usually produce large amount of acetic acid. The same for formic acid and also propionic acid. Alcoholic fermentation means that they produce alcohol. Sometimes they can also produce other kind of products as well, but alcohol is the major product. Heterogeneous fermentation means they produce everything. They produce alcohol, they produce acids, they produce lactate, they produce succinate, which is not fall under the acids or lactic acid. Enzymatic fermentation, as you can see, we got the small I don't know what is this called, because this one is quite special because this one doesn't involve any microorganism. For example, the browning of the cocoa or coffee. Okay, so this is the example of the pathways. Take note that this is only EMP and ED pathway. The phosphocatalysis pathway is not shown here because then it will look too messy for you. Okay, so you can see the numbers here. This is just the example. For example, number 1 is alcoholic pathway or alcoholic fermentation. The second one is homolactic fermentation. If you combine number 3, 2, 6, and 4, it can be mixed acid fermentation. And also number 5, I think this is alcohol. Okay, we can consider this one as alcoholic fermentation. So this is the legends. Previously, in the original lecture notes, each of these points will have 3 to 4 slides. So you have what? 1, 2, 3, 4, 5, 6. 6 points times 3 slides, about 18 slides here, but I just simplify them into these legends. Okay, so like I said to you before, number 1 is actually correspond to number 1 here. You can refer to it by yourself. Okay, just now mixed acid fermentation involve EMP pathway, but heterolactic fermentation, they are similar to mixed acid fermentation, but they are not using any EMP or ED pathway, but they use phosphocatalysis pathway. The efficiency in terms of ATP production is about half of AMP pathway. For example, if you want to produce kefir or sauerkraut, some type of fermented food, you can use this kind of microorganisms such as lactobacillus and leuconostae. That is actually heterolactic fermenters. Okay. Enzymatic fermentation is a fermentation by unorganized ferment. Unorganized ferment is actually means it is not involving any microbes. They are purely chemical reaction which is enzymatic. So for example, tea, cocoa and coffee, when we ferment them, they turn into brown by the action of enzyme polyphenol oxidase, and they don't involve any microorganism at all. Okay, so it means that this is a broader meaning of fermentation. It includes the action of enzymes so that desirable biochemical changes cause significant modification to the food. Other example can be, for example, decomposition of or inversion of sugar, conversion of starch into dextrin by similar treatment, okay, by chemical treatment, conversion of starch by enzymes, conversion of albuminous food into peptones by gastric juice, which is also the cocktail of enzymes. Okay, protein fermentation, like I said before, some people consider them as fermentation. Some people don't think they are fermentation because they don't involve any sugar. But I will be more comfortable using the word putrefaction because putrefaction is specific for proteins. Usually they will produce off odors because they produce sulfur and so on, which smell very bad, and the production of is smelling and tasting product. Okay. The action of putrefaction can be caused by proteases, peptidases, okay, some kind of amino acid. They can produce ammonia, which is pungent smell, also the sulfur component, such as rotten egg smell. Usually they are not the same as fermentation because the food is considered as spoiled already. They are not safe to be eaten. Okay. The most active proteolytic bacteria stress belong to Clostridia and okay, this is the type of bacteria that can cause putrefaction. Okay, can also be caused by bacterial peptides in human colon. Okay, in the human colon, the short peptides and free amino acid after the action of putrefaction is available for fermentation. Means that the bacteria inside our gut convert the protein into something that can be fermented. Okay, mainly for the microorganism. Okay, thank you. Later, we will go into the types of fermentation, which is, for example, continuous fermentation, submerged fermentation, and so on. Okay, I'll see you later.